Hello everyone, my name is Dr. Garry Laverty and I'm an academic at the School of Pharmacy in Queen's University Belfast. I'm the principal investigator of the Biofunctional Nanomaterials group, and I'd like to talk to you today about how we can use our peptide hydrogel platforms and also a peptide nanotube platforms for use in drug delivery and biomaterial applications. So our actual technology that we have consists of very short peptides. We call these ultra short peptides, which we can actually change the sequence of these, and we make them within our lab by chemical synthesis so that they respond to stimuli. And some of these stimuli are physiological based stimuli such as pH, specific enzymes, temperature and ionic strength. And by tailoring this self-assembly, we can control whether they form hydrogel platforms or peptide nanotube platforms in response to physiological pH. And we can also control some of their pharmacological properties, such as their ability maybe to target specific infections also. So how do we actually use these? Well, they're almost like Lego building blocks as such. We can build these materials on the nanoscale. We can modify their molecular structure as such to tailor really for specific needs that are used throughout drug delivery platforms and also throughout medicine and healthcare. One of the major focuses of our papers and publications has been using these within infections and as medical device coatings. We have also used them as hydrogel platforms in terms of wound healing, and we'll go through some of those results. And also we've looked at using them as hydrogels platforms for stem cell delivery, particularly within the dental field, which we've done collaborative work and publications with Dr. Lundy's group within the School of Dentistry at Queen's University Belfast. And perhaps one of the most funded and promising areas that we have is using the hydrogels and these nanotubes as platforms within drug delivery. And essentially that's what we're looking to do within the School of Pharmacy, is improve the delivery of existing drugs, but also of drugs that might be developed in the future. So we're heavily funded in terms of developing our hydrogels as in situ forming implants. We've also looked at using our nanotube platforms in transversing and crossing the blood-brain barrier to deliver drugs in a range of central nervous systems diseases. We've also looked at using them potentially within cancer and again to target specific bacteria, gram-positive, gram-negative bacteria, which would be implicated in a lot of hospital-acquired infections. So what are the advantages of these peptides as opposed to maybe some of the similar monomers or polymers that you would see that are used to create therapeutic platforms, therapeutic hydrogels throughout? Well, we would say that these peptides certainly are short in length, so they're only less than 7 amino acids generally in structure, which means that they actually are very cost effective to make. Now, peptide synthesis in the past was thought of as being an expensive process, but it has actually improved in the past few decades, and our capability in terms of industry is more effective at making this a more cost effective process. So they are being used more generally, and there is a wealth of peptides that are used as medicines throughout healthcare, but certainly having them as a shorter peptide sequence means that their chemical synthesis, there's less steps involved, and it's more appealing really to the pharmaceutical industry to try and manufacture these at a larger scale. Rather than say milligrams or gram scale, it's pretty obvious that these would need to be in kilograms or ton scales to get them out to patients. So we have an increased translational potential with these sorts of structures. In terms of their advantages relative to other synthetic hydrogels or other synthetic materials, the amino acid structure itself is extremely chemically versatile, and we see that in terms of how peptides are used both within nature and in the human body itself, as they form the building blocks of tissues, of hormones, and of molecules that are used throughout the body. And all of this sort of chemical versatility, functional versatility, comes from the fact that a peptide is built up of amino acids, and the R groups that go within these amino acids are very important in giving that flexibility. So this can change between each individual amino acid, and we can use this and these functional groups that are provided within this amino acid structure to actually develop our materials to very specific functional properties. Things such as the immune response to these materials are very important, so we can tailor those to what's required. We might want to see an immune response triggered in, for example, a vaccine, but we probably in most cases want to see a a lower or reduction of the immune response, and this ability to have an enhanced biocompatibility if we're trying to make materials that are injected or administered into the body. We can also tune how they degrade. So if we want to look at forming an implant that will deliver a drug over 28 days, then we want that implant to have broken down as close to that 28-day dosage period as possible. And we might want that to actually go on for longer, or we might want shorter, depending on the application that we're using. And we can also tailor the self-assembly process that we have to the pharmacological properties. So self-assembly is very much linked to, and this process of forming peptide materials is actually closely linked to pharmacological properties such as antimicrobial activity, and we'll have a brief look at that in later slides. The major disadvantage of peptides as such that exist within nature and proteins, things such as insulin, is that they have very limited stability in vivo. And from a drug delivery perspective, that means that they have to be actually probably injected intravenously, and actually being able to administer these as oral therapies is a major challenge in itself. And also in terms of having any sort of long acting or something that will stay there for a long time, like an implant, they'd be limited in terms of the use and stability with that regards as well, simply because they're recognized within the body by proteases, enzymes that attack peptides and proteins themselves, and will break them down very quickly. So our group actually looks to overcome this by developing what's called peptide mimetics. So these are molecules that are simply peptide-like building blocks, and these won't be as easily recognized by proteases within the body, simply because they have various different changes within their chemical structure that will make sure that enzymes don't actually recognize it within their active site and aren't able to break them down as readily. So what we do is actually take these structures, take some sequences. We can have three varieties which really we focus on, is the likes of a D enantiomer, which don't tend to occur naturally within the body as regularly as the L peptide enantiomer that you would see. We can also look at adding beta homopeptides, where there's an extra methylene group within each of these sequences between what's known as the amino acid R group and the carboxylic acid group. We can also form peptides, which are an increasingly exciting area of research, where the R group actually is on the nitrogen part of the amino acid sequence rather than it being on the alpha carbon, which we would generally see in nature through the L peptide enantiomer, as you can see in one here. So the peptide sequences certainly is something that is, in terms of our funded research, something that we are focused on because of its novelty as such. So when we introduce these, I will go through our peptide nanomaterials, what we are actually looking to achieve with those, and how we have developed the research, and what we're looking forward to in terms of future to try and get this sort of research translated. And then in the second part of this, I'll actually look at the peptide nanotube stuff. In terms of this is our very heavily funded research, in terms of we have support from the Research Council, the EPSRC in particular. We've had funding from the Wellcome Trust to look at these as drug delivery implants, and we've recently obtained funding from Invest ANI. So this is something that we're looking to actually produce in terms of publications in the coming months, but also to secure patent protection for, and it's something that we're very keen to reach out to industry and to expand our collaborations that we have with our existing collaborations with industry that we have. So one of the most exciting areas for this platform is actually using it as an injectable in situ forming implant. So the major aim of the World Health Organization is actually to eradicate HIV. So in terms of diseases, HIV is probably one of the most prevalent, certainly within sub-Saharan Africa, and one of the biggest difficulties and challenges is actually getting patients a supply of medication, but also getting them to adhere to medication on a daily basis. Some of the regimens in terms of HIV antiretrovirals are limited simply because they have to be taken at particular times. Multiple drugs, particular maybe 3 or 4 different drugs taken at specific times every day, and that's a huge challenge in the developed world, but also within the developing world. So the need for a platform can actually be injected and give patients that therapeutic coverage in terms of of 28 days or even longer would be of great benefit. It would be certainly something that would be increase the convenience for the patient themselves, and then hopefully we would see that in terms of therapeutic outcomes as well. Our particular EPSRC project is actually looked at developing a multi-purpose product, and simply what this means is a product where we have combined HIV prevention and also contraception within one injectable product. That currently doesn't exist, but the benefits of that would be massive, particularly within the developing world. This would provide a platform which would reduce fetal transmission of HIV, and it would also empower women within these countries to take control of their sexual health as such, and give them a discrete technology that they can use to their benefit. So our platform as such really is to give you a structural overview of this. We use our peptides, our peptides, our beta homopeptides, and a mixture of L enantiomers as well to have some sort of biodegradation within our motif to form these peptide or peptide-like molecules. So the peptide itself actually can form a hydrogel, a tissue-like hydrogel, which can be used obviously to form the hydrogel itself. But in terms of injecting that, we want a formulation that is actually fully soluble. So we add a specific group that will increase the water solubility of our peptide itself. And once this group is removed by a specific enzyme that exists under the skin, then a hydrogel will form from our peptide motif rapidly after delivery, after injection. And we can also use the chemical versatility of these peptide motifs, the different R groups and functional groups that are there, to actually covalently attach a drug to our peptide. And this is the benefits of actually increasing drug loading, and also providing a chemical attachment will hopefully reduce the ability for burst release of drugs, which we tend to see with other in situ forming implants. So in terms of our concept as such, what we have is a soluble peptide drug conjugate. We would administer that at least every month. This can be subcutaneously or intramuscularly. We tend to focus mainly for HIV use and subcutaneously, simply because in future we want patients possibly to take control of the administration of this themselves, and it's probably easier to deliver that subcutaneously rather than intramuscularly. Then there's this very specific enzyme which we look at in terms of triggering hydrogel formation. What this does is cleave this hydrophilic group from our peptide drug motif. It allows it to rapidly form a hydrogel within the skin space, and this hydrogel itself will be protease resistant, allowing the hydrogel to form and stay there within whatever the dosage period that we require. We simply aim for 28 days as a realistic goal, but this can be developed further, and we're looking at development further in terms of expanding that dosage period to months and possibly even years as such. The tissue environment in itself then, the presence of water, pH 7.5, 37 degrees centigrade, will actually hydrolyze this peptide drug linkage, and what we will get is sustained drug release from this peptide hydrogel motif. So the presence of the hydrogel itself actually reduces burst release, but certainly what we're seeing from our data is that the presence of the covalent linker that's there provides a significant improvement in terms of reducing that burst release. And the benefits of that simply is that we're trying to tailor delivery for that dosage period rather than having most of our drug delivered within the first few hours of injection. So probably the greatest question we could ask maybe at conferences is, what are the actual advantages of these? So there are certain drugs and platforms that are becoming available and are being accepted by the regulators for use in terms of HIV drug delivery and other kinds of drug delivery, contraceptive drug delivery that are out there already. So I'll go through some of the limitations that are there with some of the current long-acting injectable technologies, and how our technology actually looks to resolve that. One of the major benefits, I think, is the fact that this can be actually tailored to drugs that are currently marketed and currently exists as other administered platforms, say orally delivered drugs, but also it has the potential and flexibility to be used for drugs that will be passed in future or near future. So the fast burst release drug certainly is something that would exist with the likes of suspensions or spheres or polymer implants that are currently being used as soon as it is administered. So by actually combining our hydrogel formation and the breakage of this peptide drug bond, we're providing a covalent bond which has to be broken in order for the drug to be released, as well as the diffusion which we would see throughout the hydrogel, which will give us a significant reduction in burst release. Most drugs in terms of these other platforms tend to be physically mixed or physically encapsulated with the polymer or the implant material themselves. There's also a need probably for surgery for the likes of your polymeric implants that you would see with contraceptive drugs currently, whereas what we can do is our soluble injection, we can change the sequence and the structure of our peptide so that it breaks down into non-toxic products within the dosage interval that we require. Most of these, in terms of injectables or in situ forming implants, a lot of them, your current suspensions or nano suspensions or microspheres, actually require very large needles. What we try to do is make sure our formulation is soluble, and therefore it will allow the use of narrow bore needles because you will have a solution that you're injecting. There's also stability issues, particularly with the likes of these suspensions or nano suspension platforms. What we aim to ultimately do, and we've been discussing this with the regulators, is that we would form a freeze-dried powder that then would be mixed in a clinic prior to injection, and this provides increased stability, particularly in environments where there's temperature fluctuations. So temperature fluctuations will have a major impact on suspensions formulations, and by actually having something that's freeze-dried powder is of huge benefit in terms of stability, because you won't see your suspension flocculate or break as such, and by having a soluble injection, what we hope to be achieving is the fact that we can make this up very quickly on site because it is very soluble. There's also a limit in terms of the type of drugs that are loaded, especially within suspensions as such. So water insoluble drugs, which tend to be a lot of HIV drugs as such, but there might be an opportunity to actually expand the use of this technology. So as such, if your drug is insoluble, it will form, and it should form, a suspension in water. But if your drug is soluble, it's obviously very difficult therefore to achieve that, and you have to try and increase the hydrophobicity of the active ingredient or the drug. What we can do is we can actually precisely attach our drug to the peptide so that the peptide itself actually increases the solubility of our hydrophobic drug. We can increase the hydrophobicity of that if it's required, so that it will form a hydrogel with the water soluble drug attached. So there's a variety of ways that we can do that, and it's really down to the ability to tailor that peptide sequence to the requirements of the drug that we need. There's also this ability that for particularly antipsychotic and illnesses such as schizophrenia, where they actually form oily injections which are injected intramuscularly, these tend to be very, very oily as such and hydrophobic in nature, which simply then, once it's injected intramuscularly, will cause side effects and persistent pain, sometimes lasting for months in patients who are administered that platform. For the likes of ours, we will always kind of stick to this aqueous platform as a water-based solvent. It shouldn't be oily, therefore it shouldn't actually have this persistent pain hopefully in place because of its improved biocompatibility of the solvent water itself. What are the other potential uses of our peptide hydrogels? Well, in other applications, things such as any disease with medication adherence issues. So we've covered the likes of HIV, AIDS, combining that with contraceptive cover, but also we've done some studies in terms of their use in schizophrenia and also substance abuse, so things such as alcohol and drug dependence. We've also looked at their delivery in terms of intratumoral injectables or in situ forming implants, where we can deliver drugs to the tumour site. Possibly we could introduce radiosensitizers to improve the outcomes that are associated with radiotherapy. And we've also looking at using these in terms of platforms for difficult to deliver sites within the body. So the likes of ocular delivery, spinal delivery, the delivery to the central nervous system as such is of massive potential. There's also ability to use this platform as a vaccine. So peptides have been shown in research to be used as immune adjuvants, as opposed to things that are commonly used now, such as Illumina. our platform as a hydrogel implant may actually provide the benefit for extended protection and possibly elicit an immune response which is more long lasting when we use a peptide rather than other types of adjuvants. And also we're looking and have a history of looking at these hydrogel and nanomaterial platforms, both of them in terms of infection, and that will become the focus of the next few slides. One of the major areas where our peptides actually have potential is in combating antimicrobial resistance. So we're all very much aware within the public and as scientists that there's a major problem in terms of superbugs, infections, getting and developing new drugs to counteract those, and also how we actually use existing therapies more properly. So one of the major problems in terms of superbug infection in hospitals especially is through infections of medical devices, things such as catheters, hip replacement, heart valves, which we tend to see in elderly people to help them support their lives. And these actually form an increased source or reservoir of superbug infections simply because the implant surface itself is implanted within the body, and infection or bacteria, microbes can actually attach to those and form a very slimy biofilm infection, which tends to have increased tolerance to antibiotics or therapies that we currently use. So we're trying to have to increase the concentration of drug that's administered, which will lead to increased resistance possibly if the bacteria are able to tolerate those increased doses because of the kind of barrier that this slimy biofilm provides. So it's a big burden in terms of society, in terms of healthcare budgets, patient morbidity, mortality, and a lot of research groups have looked at actually using hydrogel coatings to try and prevent these infections and these biofilm developing within the surface. So there is a body of research that we have looked at to actually use our hydrogels potentially within this field. So one of the work that we actually conducted, which was published in 2014, was to use a very short peptide sequence, and I had the opportunity actually to go to Brandeis University in Waltham to go to Professor Bing Zhu's group, who is a world leader in terms of the use of these peptide materials. Within the School of Pharmacy, we had done a lot of work previously with regards to antimicrobial peptides. So we did find that we could actually utilize Bing's short peptide hydrogel forming motif and try and modify it in terms of making it antimicrobial. So as you can see from some of the images that we have here, what we simply did was add cationic, positively charged amino acids to his naphthalene diphenylalanine motif in order to make it more cationic. And by doing this, what we hypothesized was that it would increase the ability of the peptide and the peptide hydrogel to actually attack the negatively charged anionic cell membranes of bacteria and target those selectively because they tend to be negatively charged, that as opposed to mammalian cells, which tends to be more of a neutral charge overall. And it's something that is used in the antimicrobial peptides field regularly as a target. We looked at simply just looking at their fibrous structures in terms of electron microscopy, and really what we wanted to look at was their ability to disrupt biofilms or target biofilms themselves. So whilst I was in Boston, we did look at developing these, and I brought some of the samples back to the Belfast with me, and we looked at targeting bacteria that would be common within medical device infection, and one of those is Staphylococcus aureus, which exists within the skin, but when it has an opportunity to actually become more pathogenic, it will take it, so it will attach to the surface of an implant and form these biofilms. As you can see with the example of Pseudomonas aeruginosa here, it does the same thing, mainly in urinary catheter infections. You would see Pseudomonas, and we had studied it as well. So one of the trends that we are seeing here, we grew our biofilm simply within our lab, and we put hydrogels above that to see would it target it within a 24-hour time period. And what we've seen was that there was a reduction, certainly in most cases, once we added a cationic species to our naphthalene diphenylalanine. There was a reduction in biofilm formation of above 80%. And the lysine, the KK, actually seemed to be the most beneficial in terms of antibiofilm properties. We also looked at using these within in vivo assay, and one of the more promising assays that we use within the School of Pharmacy is a waxworm assay using Galleria. We can actually inject these Galleria both with infections and bacteria such as Staphylococcus aureus, and inject them with our peptide as well to see if it reduces the incidence and the amount of bacteria that are present, and also if these are, in terms of platforms, gives us an overview of whether there's any sort of toxicity issues with these peptides themselves. In very simple terms, we can test over a few days whether they form live or dead in terms of the Galleria, and it's a good visual aid in terms of toxicity. But we can also extract the lymph that exists within these Galleria to get some idea as to the quantity or the colony forming units that exist of these particular bacteria. So our study in itself, we just simply administered our most promising peptide, the dilysine variant of naphthalene diphenylalanine. We inoculated these Galleria with different types of pathogens that also exist again in medical device infection. What we tend to see actually was that we had a reduction in Staphylococcus aureus, but we couldn't actually achieve that certainly in the in vivo model with our Staph epidermidis, which is related to Staphylococcus aureus. They're both gram positive, and also the gram negatives just tended to be more challenging, but we did get a reduction with Staphylococcus aureus as such. These results were published, and the assay itself was a new assay that we developed that would be of use to any sort of researchers that are looking at anti-infectives. So when we think about the potential use of these platforms, well, certainly as coatings, if you think about the biomaterial industry, the medical device industry, it's estimated, and I would say this is a lower estimation than currently exists, because this data is from probably 10 years ago, is that there's a $75 billion industry worldwide, and that's down to really an increased age demographic throughout the developed world. So we can use these things for urinary catheters. They tend to be infected by E. coli, Pseudomonas, Proteus mirabilis. Intravascular catheters, which tend to be infected mainly by Staphylococcus aureus and Staphylococcus epidermidis, gram-positive infections. Hip replacements as well, and cardiac devices. So if we can actually provide a means to prevent infection and biofilm formation occurring, that would be of massive benefit. We can also use them possibly as hydrogel really to extend the delivery of currently administered antibiotics, and that would be a benefit just to make sure that we are using our current formularies of antibiotics as wisely as possible. Gram-negative infections in particular are a massive problem in terms of developing new drugs for, so using existing therapies and extending their use would be of great benefit. We can also provide, again, extended or prolonged release of these drugs that currently exist in antibiotics. And the major thing is that we're trying to actually translate these into the platforms that would be of benefit to patients. When we think about the use of peptide hydrogels, probably the easiest formulation to actually develop as such would be the likes of a wound healing therapy. So using our hydrogel to support the development and growth and regrowth of tissue and allow the body to heal itself. So one of the major probably areas in terms of wound health would be the likes of chronic wounds, things like diabetic ulcers, which are unable to heal fully themselves or respond to any sort of treatment within a 4 to 12 week window. And this creates not only issues in terms of patient suffering, but also problems in terms of the money that is spent in trying to develop these therapies, and also the use of current therapies and wound healing. So there is certainly an avenue here for using our peptide hydrogels to try and improve patient health in wound healing, especially chronic wounds. So the chronic wounds tend to actually differ from acute wounds, really, that have this prolonged inflammatory response where the body is kind of attacking the wound itself and is preventing it from healing, and there's a lot of inflammation that's around wounds, whereas the inflammatory process in acute wounds actually tends to be a more beneficial thing to try and prevent infection and increase healing within the first few days of a wound occurring. What they're actually finding is that the benefit exists for the use of non-steroidal anti-inflammatory drugs, things like ibuprofen, naproxen, within chronic wounds themselves. So what we had was actually an idea where we could attach our non-steroidal anti-inflammatory drugs to our peptides and try to form a multifunctional peptide hydrogel platform, which would, yes, form a hydrogel, which would be like tissue itself. It would be very biocompatible because it's a peptide based system. It could also be antimicrobial, as we've seen in the results that we had with our dilysine variant. And we also, by including a non-steroidal anti-inflammatory drug, we can reduce inflammation in the chronic wound by delivering that drug as well. And we also wanted to look at increasing wound healing as such by increasing angiogenesis, so this idea of a pro-angiogenic motif. We have published within this area, as you can see in our publication below from 2016, but we are working towards going forward and certainly proving in terms of the pro-angiogenic response of this. We have developed a heparin mimetic motif, and heparin would tend to have been used in research to improve angiogenesis and the healing of wounds, and we've also looking to maybe show that it does specifically target inflammatory response. So we've shown already that it actually has selective COX-2 inhibition once we add naproxen to our peptide motif. We've shown that it's biocompatible, antimicrobial properties exist, which we'll look at in the next slide, and we just need to kind of prove that it has other inflammatory mediators. The likes of your NF-kappa B has been shown to be involved in chronic wound health, so trying to inhibit that would be of major benefit, and also the likes of your toll-like receptors, which tend to upregulate the immune response by binding to different kind of biomolecules, DNA, which might be released maybe by damaged tissue cells themselves from the host, or also DNA or bacterial lipopolysaccharides that might maybe be released from damaged bacteria. The idea that we have is actually very much shown in terms of this traffic light system. The red is where we're still currently working on, and we're kind of hopeful of getting maybe some industrial engagement with this as well. When we look about the properties of this gel, we did the exactly same thing in terms of antimicrobial activity. The antibiofilms we looked at themselves, and we also looked at the response against planktonic or liquid bacteria. Simply our biofilm model, we were getting a biofilm reduction, certainly for most of these dilysine motifs when we added an NSAID, the likes ibuprofen, indomethacin, naproxen, we were getting above 90% for most cases in reducing, in this case, Staphylococcus aureus. But the biofilm model itself was actually limited in terms of it was only giving us a percentage reduction, and what we really needed was an idea of colony count formation. So what we did do was actually have a mixture of biofilm cells and these liquid planktonic cells, place our hydrogel within tissue culture plates, and what we did was inoculate them with different types of bacteria and then take samples to establish the colony forming units, the exact amount of bacteria that was growing there and that was viable. So it gives us like a log reduction, which is more acceptable scientifically in terms of showing how much we can reduce bacterial load at the infected wound site. It's also a case that, you know, in the variety of collaborations and research that we've looked at, that you actually don't want to totally make the wound environment sterile, so a reduction is more preferable, but certainly we need this idea of log reduction rather than just looking at percentage reduction alone. If you think even about bacteria and how antiseptics are kind of generally marketed, it's all about getting 99.9% reduction of bacteria and kill, and it's very difficult to achieve that within our biofilm assays simply because it's more of a qualitative assay. We're using things such as Almar Blue cell viability reagents as a comparison really to biofilm growth controls without our peptide in order to establish how much biofilm reduction there is. So we are kind of limited by that, so I would always recommend that we have this mixture of biofilms and liquid bacteria to give us our colony forming units, to give us an idea of the efficacy of our platform. And certainly results as they stand are very, very promising in taking this NSAID peptide hydrogel forward within the naproxen variant, particularly beneficial in my view. If you have an interest in our peptide hydrogel work, and particularly those that are related to their use as antimicrobials, there is an expanding field in terms of linking the self-assembly process, how these actually form hydrogels with antimicrobial properties. And we have actually published this as a review in the Soft Matter journal. We were invited to submit in terms of the Emerging Investigators issue in 2021. So I think that does provide a good idea of the future perspectives of this field and the promise that peptide hydrogels and our nanotube platforms, which we will go on to look at, have in terms of providing an alternative platform to resolve the increased burden of antimicrobial resistance. So now I'll take a look at the peptide nanotube platform that we have, and a lot of this work has also been published. Our initial funding in this area was actually provided by the Royal Society, and hopefully we'll be able to differentiate the difference between our peptide nanotubes and our hydrogel platforms. The chemistry and the physical forces and intermolecular bonding and such that are involved are very similar between the two platforms. It's just really that the peptide sequence is what's different, that will determine whether it forms a hydrogel or these nanotubes. It tends to be an alternative platform as opposed to something like carbon nanotubes. We would say that our peptide again is more chemically versatile. Our R group will allow the attachment of specific drugs or entities that we think have a medical benefit, whereas bonding to a carbon nanotube and the bonds that are involved in those tend to be a bit more difficult. So the first thing that we wanted to do actually was develop these platforms and based on the minimum motif that was present. So we chose the diphenylalanine motif, and what we did was actually just alter an A, as you see here. This is an L enantiomer version of it with a carboxylic acid group attached. B, we had a D enantiomeric variant, the D isomers that were stereoisomers involved, and what we had was a carboxylic acid group for it. And then simply for C, what we did was just modify that to have an amide group at the end as well. We wanted to see if that had any sort of significant difference in terms of how these were able to self-assemble into nanotubes, or whether any sort of differences in pharmacological activity. What we seen was that these were capable of forming your nanotubes, as you can see in the SEM images, at around 1 milligram per mil, and these structures tended to exist mainly within a diameter of the nanometer range once we formulated them and allowed them to self-assemble overnight. Once we had proven the ability of these nanotubes then to self-assemble, what we wanted to do was explore their ability against biofilm forms again of medical device related pathogens. So simply what we did was initially look at how under a microscope using SEM analysis how these peptide nanotubes impacted the cell membranes or the structure and architecture of bacteria. As we can see in images A and B, both and all of these are published within our recent paper in Active Biomaterial. What we seen was that they were forming like with pores within the cell membranes of these bacteria, and especially within gram-positive bacteria. What this actually led to was kill of viable bacteria cells and reduction of infective cells as such. In gram-negative, such as E. coli and Pseudomonas aeruginosa, there was pore formation in the cell membranes. It just weren't to the degree that we actually correlated the inhibition or kill of the bacteria themselves, and these were all over 24 hours. What we kind of put this down was probably an ability of these self-assembled nanotubes maybe to create ion channels possibly, or surfactant-like action on the cell membranes of the gram-positive bacteria in particular. And this is something actually that you see in amyloid plaques as well. There's an increasing growth in research within amyloid plaques and how potentially they could be linked to infection as such, and the body's ability to try and generate a response to infection in the central nervous system, which then maybe leads to Alzheimer's. So we looked at that in greater detail within our soft matter review, but certainly there is an ability to look at this self-assembled nature of peptides, link it to amyloid plaques, and potentially this could be one way in which they are trying to attack bacteria as such. Maybe the reduced ability against gram-negatives is due to the polyanionic, so the very kind of negatively charged nature of the biofilm forms of gram-negatives. Alginate is present within Pseudomonas aeruginosa biofilms. Colonic acid, they're very polyanionic, negatively charged, and they actually might prevent a diffusion of these carboxylic acid terminated diphenylalanine molecules, which tend just to be more negatively charged. But really the research space within this simply has to be looked at and explored in greater detail by ourselves and other groups. We can also see there was a difference in terms of the concentration. So once we increased the concentration, in this case of our diphenylalanine with the carboxylic acid attached, the L enantiomer, we've seen that there was certainly a reduction in the presence of the bacteria that was attached to the surface of our Calgary biofilm device plates that we had here, the pegs of those. So certainly there was an ability for our peptide, as we increase the concentration, to kind of eradicate existing biofilms, and you can see this from D to F in our images, whereas the controls with no peptide present, you can see very clearly that there is surface attached biofilms present. We also have studied their use through Royal Society funding to try and use these nanotubes. Like many nanoparticles, carbon nanotubes, they've been widely used to try and deliver drugs across biological barriers. One of the major benefits of peptides, again, is their chemical versatility. So as opposed to something like carbon nanotubes, it should be readily easy to actually attach drugs to a peptide, or attach kind of any sort of therapeutic molecule of interest. What we aim to look at is certainly their ability to cross the outer membrane of gram-negative bacteria, because simply what this could do is allow an avenue to encapsulate or incorporate existing antibiotics which only have activity against gram-positives that aren't able to cross the outer membrane of a gram-negative bacteria in order to exert their effect. So if we could help to actually shuttle those across, they could potentially expand the use of currently existing and licensed antimicrobials or antibiotics for use in gram-negative bacteria. One of the major issues is obviously the blood-brain barrier in terms of a biological barrier. So we have looked at using these in terms of encapsulating and incorporating drugs that would be used within diseases such as Parkinson's disease, and also HIV in terms of central nervous system reservoirs of HIV infection. It's very difficult to treat those too. And also intercellular cancer cell delivery, trying to deliver payloads or therapeutic payloads that can target the nucleus of cancer cells in order to exert their effect as well. So simply we're trying to use the peptide nanotubes as a means to shuttle drugs across these barriers to increase their therapeutic efficacy. Within our active biomaterial paper, we actually did perform an assay using the fluorescent molecule 1-phenylnaphthylamine, NPN, and this is used as an uptake assay really, and I can show the permeability of the outer membrane when we use something like polymyxin B, which readily crosses the outer membrane of gram-negative bacteria. We can use that as a control, and what we've seen was that our peptides, both the D and L forms of the carboxylic acid diphenylalanine variant, were both able to cross the membrane at concentrations of 5 milligrams per mil and above, as readily as polymyxin B as such. So the actual formation of these pores as such is probably helping that we've seen in the SEM images earlier, and there is the potential probably to mix these nanotubes and encapsulate them with drugs that are used in gram-positive bacteria to allow them to cross the outer membrane and to use alongside our own peptide nanotube platform. With regards to the blood-brain barrier, what we had hypothesized was that we would be able to target 2 routes across the blood-brain barrier using these diphenylalanine peptide motifs. Because they contain diphenylalanine, these are very lipophilic molecules, so as such they should actually be able to target what's known as the transcellular lipophilic pathway of blood-brain barriers, and able to shuttle drugs across the astrocytes and into the brain as required. We've also looked at things such as absorptive endocytosis, which have been previously shown to be of benefit for anything that forms like a nanotube structure, like carbon nanotubes. They tend to have this ability to be taken up and allowed to cross the barrier by this energy dependent pathway, and we have looked at that in greater detail. We've looked at using these in brain tumours. So the blood-brain barrier in brain tumours is actually compromised as such. So it doesn't form as much of a barrier, but it still exists in terms of providing a barrier to the therapy and therapeutic efficacy of anti-cancer drugs. Particularly in brain tumours, there's a high need for new platforms to try and improve the ability of chemotherapy drugs to target the tumours, especially the likes of glioblastomas. So this did form an avenue both for our grant funding and also for papers that we've got from that, in order to try and use these peptide nanotubes as a means to cross this barrier and allowing cancer drugs to be delivered to really only the cancer cells themselves rather than healthy human cells. There is the potential as well to kind of modify our peptide and our peptide nanotubes to introduce ligands so that they will only target tumour cells. But again, it's something that we are looking at for future, and we probably would need specialisation with somebody who is able to do that in terms of the cancer field. And linking in terms of collaborations with that. We also looked at the toxicity of these. We did various cell viability agents such as MTS, and we did hemolysis assays with these. And overall, the trend that we actually found with these was that both our carboxylic acid diphenylalanine variants were actually very non-toxic as such, but once you added that amide group at the end, as you can see here in the blue, that was our C molecule, it actually did increase the toxicity as such, probably because it's more cationic and it's more likely to kind of target cell membranes as such and break them down. So that was a trend that we had seen that probably had more negatives in terms of our amide terminated sequence than it had positives. Okay, it was probably had an ability to potentially be an anti-tumour platform itself and to kill cancer cells, but also it would have toxicity against our subcutaneous cell line, the NCTC929 cell line, which tends to be used by medical device and drug developers as a reference in terms of mammalian cell viability and toxicity. But we were very pleased in terms of there was no significant toxicity up to 10 milligrams per mil for diphenylalanine carboxylic acid, both the L and D variants of those. What we had seen then, we had actually tested toxicity again using live dead staining and also our MTS against glioblastoma cell lines, and this was published in 2020 through a paper, as you can see below. And what we found was a similar pattern that we had for our subcutaneous NCTC 929 cells that we had in our previous slides, that the amide terminus NH2 terminated diphenylalanines were simply killing our cancer cells at 10 milligrams per mil, whereas our carboxylic acid terminated platforms weren't. I would say that the benefit and most potential is going to come from our inert diphenylalanine nanotubes, the carboxylic acid variants, simply because then we can use those to encapsulate our cancer drugs, and they will provide the anti-cancer activity. It's a bigger challenge, I think, to use our toxic amide terminated diphenylalanine peptide nanotubes, simply because they probably will therefore be toxic against human mammalian cells as such. And if you think about how we're going to deliver these, we're probably going to have to inject them intravenously so that they're delivered to the brain. Trying to inject those locally into the brain is going to be extremely difficult, and so therefore you need them to be as biocompatible and non-toxic as possible. But within this publication, what we actually did was load them with a fluorescent dye. We wanted to see how drug was going to be loaded within this and how this dye would actually be released. We wanted to maybe look at changes in pH. The pH of the tumour environment tends to be more acidic, and we want to see if actually our peptide nanotube would actually unravel within a more acidic environment. We didn't actually achieve that or see that, but we did see using some of these images just where our drug is loading. We're getting very good drug loading above 80% in all cases. So in terms of a platform, in terms of loading drugs and trying to deliver those, it has huge potential for future use, but certainly there is a lot of challenges to overcome with the use of peptide nanotubes, more than probably that we have with our in situ forming hydrogel implants. And as such, it's probably not a surprise that that the use of our hydrogels is probably the major focus of our work going forward, whereas these we are still working towards, but in terms of clinical development we are probably behind our hydrogel platform. So in terms of the nanotubes, the challenges that are there to overcome is still this reliance on an enhanced permeation and retention effect, especially within the tumour environment. So we're relying on tumours having an increased blood supply in order to uptake these nanotubes with a drug attached, rather than having any sort of specific trigger that's involved that will allow it to deliver within a cancer environment. So the likes of your physiological trigger, something that would be ideal is we have looked at trying to trigger the unraveling of our nanotubes in terms of hypoxic low oxygen environments which exist within tumours. We've attempted to show unsuccessfully, I would say, as shown in the previous slide, a pH trigger, but we could develop our platform, our nanotube platform, further to have a pH trigger within it. Something like histidine is pH responsive, so including that as an amino acid within our secrets might be a way forward. Also, looking at different proteases, MMP proteases possibly that are present in increased amounts in the tumour environment might be another way forward to try and unravel and deliver our chemotherapy drug or whatever we're aiming to use to actually improve and to kill the tumour cell. We also can make sort of chemical changes. So I had spoken about ligands, maybe adding these short peptide motif would be a huge challenge because it's going to affect probably its ability to form a nanotube as such. What we might look towards is actually increasing our sequence of our peptides. There are octopeptides which form from nanotubes and are built of 8 amino acids, and they probably will be able to incorporate a ligand or a short peptide motif which will help tumour targeting more than a dipeptide motif. And also the size. So a lot of our studies were performed after 16 to 24 hours after we had actually made these peptides, and that is a realistic timescale in terms of formulating these and using them. But as we leave those, and we've shown that after a week or two of forming these, they actually go to make more of a macromolecule as such rather than a nano molecule on a nanometer scale. And this is a big difficulty for trying to maybe deliver these into cells themselves, into the nucleus as such, because they're just bigger than the cell itself. So trying to prevent that self-assembly process is a big challenge in terms of intercellular delivery. So hopefully today's talk has helped to inform you of some of our recent work and some of the work that we've performed obviously in the past. I'd like to thank our funders first of all to allow us to do this work. Everyone from the MRC and EPSRC, the Wellcome Trust, and other funders that we have. Also those within our own group, as you can see. If you have any further interest in our work, please do feel free to email us, or you can check us out on Twitter, and we also have a group website as well. I would also like to thank our collaborators who are directly involved in the work that's here. The Zoo Group, who hosted me in 2013, allowed me to perform and learn a lot in terms of this area of research, and also the Atoms Group, who we closely collaborate with the School of Chemistry and University of Glasgow. Thank you.